If you are listening to this right now, you are deep in the trenches preparing for your board exams, likely the FRCS in plastic surgery, and you need high-yield facts delivered with absolute precision.
Exactly. We are stripping away the fluff today. This is purely about the architecture of the human upper limb.
Right. Our mission for this deep dive is a strictly factual, highly targeted review of the embryology and morphogenesis of the upper extremity. We're moving straight from early developmental timelines all the way down to, the specific molecular mechanisms and signaling pathways.
Yeah, because understanding the structural sequence, the vascular evolution, and those critical genetic axes is, well, basically the only way to figure out how an undifferentiated mass of cells actually forms an arm.
So true. Now, before we start the clock on the actual anatomy, do yourself a favor and press follow on the show right now so you don't miss a single review session during your prep. And if you learned something valuable today that secures a concept for the exam, leave us a rating. And for those of you who need to go even further, you should definitely get a premium subscription to the show. That premium feed is where you'll find our exclusive journal club and clinical cases deep dives.
Highly recommend that for board prep.
Okay, let's unpack this. Take us back to day 26. The embryo is developing, but there's no arm yet. How does the upper limb bud initially form, and what are the key milestones leading up to week 8?
Right, so at that day 26 mark, the upper limb bud first appears in the flank of the embryo. It's deceptively simple at this stage. It basically consists of a core of lateral plate mesoderm, and that core is covered by a thin outer layer of ectoderm.
And for the sake of the boards, we need to be crystal clear about the destiny of that specific core tissue, right? The lateral plate mesoderm.
Yes, that is vital. That lateral plate mesoderm is the foundational building block for the upper extremity's rigid structures, so it's destined to differentiate into the bone, the cartilage, and the tendon.
Just those three.
Exactly. Now, if we look at that outer shell, the ectoderm, something highly specific happens at the very tip of the bud. It begins to thicken along the anteroposterior axis.
The anteroposterior axis in the embryo being analogous to the radial ulnar axis in the mature arm, correct?
Right, that's a vital translation to keep in mind for clinical questions. This thickened region of ectoderm at the tick forms a structure called the apical ectodermal ridge, or the AER. We'll spend a lot of time on the AER later because it is the primary engine driving the limb outward.
Got it. So by day 26, we essentially have a tiny bud with a specialized tip, but a bud is a long way from a functional hand. Moving down the timeline, what's happening at day 33?
Well, by day 33, that initial bud has flattened and expanded into a paddle-shaped hand, but obviously a paddle made of soft tissue needs an internal frame.
Right.
So inside that paddle, we start to see prechondrogenic condensations of mesenchyme.
Basically, the biological blueprints outlining exactly where the skeleton will eventually lay down bone and cartilage.
Exactly. You've got the blueprints at day 33, but structural scaffolding isn't enough, An arm needs to move. And we established that the original core, the lateral plate mesoderm, only handles bone, cartilage, and tendon.
Which brings us to a major shift at day 36. Where does the muscle come from?
This is a massive concept for the exam. The muscle does not originate from the initial bud. At day 36, nerve trunks begin to enter the developing arm, and a completely new tissue-type Semitic mesoderm actively invades the limb.
Oh, wow. It's almost like a secondary construction crew arriving on site just to handle the motor elements.
That is a highly accurate way to look at it, yeah.
Yeah.
There's a strict division of labor. The invading Semitic mesoderm aggregates into very distinct dorsal and ventral masses within the limb.
Okay.
And once situated, those masses differentiate into myoblasts, which ultimately mature into the muscle tissue of the entire upper limb.
So if a board question asks about the embryological origin of, like, the biceps or the triceps, the answer is the invading Semitic mesoderm at day 36.
Absolutely.
Absolutely not the lateral plate mesoderm.
Right, never the lateral plate.
All right, let's fast forward a bit. We have the blueprints, we have the invading muscle cells. By day 42, the digital rays, the precursors to the fingers, are present, giving the hand a distinctly webbed appearance. Then we hit the crucial 7th week.
Yeah, the 7th week is characterized by massive spatial organization. First off, the upper limbs undergo a 90-degree lateral rotation.
Right, so the thumb essentially rotates from pointing cranially to pointing laterally, bringing the palm to its final anterior-facing position.
Correct. And this isn't happening in isolation. While the limb is rotating, the elbow joint begins to flex. Simultaneously, actual ossification, The hardening of those initial blueprints into true bone kicks off.
Oh,
And perhaps most visibly, the digital separation finally occurs, turning that webbed paddle into distinct fingers.
I want to spend a second on that digital separation process because it's a bit counterintuitive.
Oh, definitely.
So it's less like building a hand by gluing fingers together, and more like a sculptor taking a solid paddle of clay and carving out the negative space, the apoptosis, to reveal the fingers.
That's spot on. The biological chisel doing that carving is apoptosis. It's the highly regulated, programmed cell death of the tissue situated directly between those digital rays.
So the tissue isn't stretching or moving out of the way.
No, it is actively dying off to create the gaps. And because of this incredibly rapid sequence of rotation, ossification, and apoptosis, by the time the embryo reaches the 8th week, the upper extremity effectively resembles a miniature adult upper limb.
That's wild.
Mm.
From a tiny bud of undifferentiated lateral plate mesoderm on day 26 to a miniature, fully separated adult limb by week 8.
It's a staggering pace.
It really is. Okay, let's pivot to the plumbing required to sustain that pace.
Yeah.
Because obviously a structure growing and differentiating that fast needs a vascular supply that can keep up. How does the body establish this perfusion?
Well, the initial blood supply to the early limb bud is remarkably rudimentary. It's just a simple, disorganized capillary network. Okay.
But as the structural development accelerates, this network has to organize. It coalesces to form a main stem artery that pumps blood in, which subsequently drains into a marginal vein.
And those two vessels establish the permanent major vascular axes of the mature arm, correct?
They do. The main stem artery evolves into the subclavian axillary brachial axis, and on the venous side, the marginal vein becomes the basilic axillary subclavian axis.
Tracing that arterial axis further down past the elbow is where we see a very dramatic and highly testable shift in dominance. The brachial artery branches into the interosseous and median arteries. But the median artery plays a very bizarre temporary role in the embryo, right?
It's a fascinating quirk of human development. Initially, the median artery provides the absolute main blood supply to the developing hand. It's the dominant high-flow vessel.
Right.
But it only holds that title for a brief window. Around day 44, it's usually replaced by the ulnar and radial arteries, which rapidly mature and take over the primary perfusion of the hand. I'm trying to wrap my head around why the body builds a major artery just to replace it a few days later. It's almost like building a massive starter highway just to handle all the heavy construction traffic for the hand initially. But once the massive radial and ulnar freeways are built, it just gets downgraded to a local access road strictly serving the median nerve.
Exactly. Because it doesn't just vanish into thin air, it regresses. It shrinks down from being the primary supplier of the entire hand to a much smaller, highly specialized vessel.
Just for the nerve.
Just for the nerve. This highlights a broader principle in embryology. Honestly, transient structures often adapt their function as the surrounding anatomy matures.
Okay, so we've covered the timeline from day 26 to week 8, and we've traced the evolving vascular axis. Now, let's shift from what happens to why it happens. We have these undifferentiated cells dividing rapidly. How do they actually know where to go?
Right, the patterning.
Yeah, like how do they know to become a shoulder instead of a fingertip, or a thumb instead of a pinky? Let's think of the developing limb bud like a complex 3D printer. To print the arm correctly, the cells need exact coordinates in 3 dimensions, x, y, and z.
I like that analogy.
Let's start with the first coordinate, the proximal-distal axis, the line from the shoulder down to the fingers.
So much of our foundational understanding of these coordinates actually comes from incredibly precise tissue transplantation experiments within chick embryos.
Oh, right.
Yeah, the proximal-distal axis controls how the limb differentiates into a shoulder proximally and fingers distally. The primary control center for this entire axis is the AER, the apical ectodermal ridge, which we noted forms at the very tip of the bud on day 26.
Here's where it gets really interesting. The chick embryo grafts. How do those experiments prove that the AER is the control center for shoulder to finger length?
Basically, by physically altering the AER and observing the result. If a researcher performs a resection of the AER in a chick embryo, it predictably results in a truncated limb, and the timing is perfectly linear.
Meaning?
Meaning an earlier resection results in a much more proximal limb truncation. If you remove it very early, you might only get a humerus. If you remove it later, you might get a forearm but no hand. Conversely, if you graft an additional AER onto a limb bud, it elongates the limb.
Okay, so the AER is clearly the engine driving the limb outward. What is the specific molecular fuel running that engine?
The AER produces a family of proteins called fibroblast growth factors, or FGFs. For board preparation, you need to memorize the most critical ones. FGF2, 4, and 8.
FGF2, 4, and 8. Got it.
Yep. These FGF signals are secreted by the AER, and they command the underlying mesoderm to continuously proliferate and extend distally.
That locks in our first coordinate. The proximal distal axis is controlled by the AER pumping out FGF2, 4, and 8. Let's move to the second coordinate on our 3D printer, the anteroposterior axis.
Right.
This is the axis that ensures an ulna and a little finger develop on one side of the hand, while a radius and a thumb develop on the other. What's the control center dictating the width and identity of the digits?
The critical area for the anteroposterior axis is a structure called the zone of polarizing activity, or the ZPA.
The ZPA.
Yes, the ZPA is a very specific concentrated cluster of mesenchymal cells located strictly in the posterior margin of the developing limb bud.
And what happens if we manipulate the ZPA in those same chick embryos?
The results are visually striking, honestly. If you take a graft of ZPA cells from the posterior margin and transplant it to the anterior limb margin of another embryo, it actually induces a complete functional mirror image duplication of the digits.
Wow.
Yeah, you end up with a limb that looks like two hands fused together at the thumbs.
That's crazy. Okay, so if the AER uses FGFs to communicate length, what molecule is the ZPA using to dictate this incredibly potent thumb-to-pinky map?
The ZPA produces a specialized protein called sonic hedgehog, abbreviated as SH. The entire anteroposterior identity of the limb is dependent on the concentration gradient of sonic hedgehog.
A gradient, okay.
Right. Higher concentrations of SH predictably result in more posterior or ulnar structures being formed. So the cells located closest to the ZPA in the posterior margin receive the highest, most concentrated dose of SH and are instructed to become the pinky and the ulna.
And the thumb?
The cells furthest away on the anterior margin receive little to no SH, and that lack of signal instructs them to become the thumb and the radius.
Okay, so we have the AER driving length using FGF, and the ZPA driving width and digit identity using the sonic hedgehog concentration gradient. Wait, I'm stuck here.
What's up?
If the AER is just driving length and the ZPA is just dictating pinky to thumb identity, why does a clinical issue with one usually ruin the other? Shouldn't the ZPA failure just give you a weirdly shaped but like normal length hand? I'm struggling to see why these are taught as interdependent systems.
It's an incredibly common point of confusion, but grasping this is essential for understanding clinical anomalies. They are fundamentally interdependent because of a biological feedback loop.
A loop?
Yeah. The FGFs produced by the AER are absolutely required to trigger and maintain the expression of sonic hedgehog in the ZPA. But it goes both ways. The sonic hedgehog signal from the ZPA is simultaneously required to maintain the structural integrity and function of the AER.
They feed each other.
Exactly.
If an embryo loses the AER, the ZPA loses its supply of FGFs and immediately stops making sonic hedgehog. And if an embryo loses the ZPA, the AER loses its sonic hedgehog signal, it falls apart, and it stops producing the FGFs needed for length.
That is the exact mechanism. This interdependent loop perfectly explains why a localized embryonic insult rarely affects just one isolated axis. It's the reason why a clinical loss of limb elements almost always occurs in both the anteroposterior and proximodistal axis simultaneously. If one control center fails, the feedback loop collapses, dragging the other down with it.
If you're taking notes, circle that feedback loop. It is the kind of high-yield synthesis that separates a passing score from a failing one. All right, we have our X and Y coordinates. Now we need the final Z-axis of our biological 3D printer, the dorsiventral axis.
The third dimension.
Right, with the shoulder to fingertip and thumb to pinky axes locked in, how does the limb bud distinguish the dorsal surface, the back of the hand, where the fingernails grow, from the palmar ventral surface? We haven't talked much about the tissue layer covering the rest of the bud.
Well, the critical area controlling the dorsiventral axis is the non-AER ectoderm. This is the general ectoderm covering the entirety of the limb bud outside of that specialized tip. And once again, chick limb bud experiments give us the answer.
Oh, the chicks again.
Always the chicks. If a researcher removes the ectoderm from a chick limb bud, intentionally reverses its dorsiventral orientation and replaces it, it will produce a complete reversal in the polarity of the underlying mesoderm. The ectoderm essentially dictates the architectural identity of the deeper tissue beneath it.
So what does this all mean? Let's get down to the molecular signals. What is the specific genetic instruction for the dorsal side?
Dorsal identity is actively controlled by WNT7A. WNT7A is a signaling protein encoded by the WNT7A gene. This gene is expressed specifically and exclusively by the dorsal limb ectoderm.
Okay, 17A on the back.
Right. Its presence dictates dorsal limb identity, instructing the underlying tissue to form dorsal structures like fingernails and extensor tendons. Want7A achieves this downstream by activating the expression of the limb homeodomain factor, LMX1, within the underlying mesenchyme.
Let me make sure I have this sequence. The WNT7A gene makes the WNT7A protein in the dorsal ectoderm, which then activates LMX1 in the mesoderm to build dorsal structures.
Precisely.
But what stops that signal from just wrapping all the way around the finger? I visualize this, 17A, and the ventral signal as a literal turf war. 17A wants to paint the entire hand with fingernails, but the ventral side has to have some kind of active firewall to block it.
That's exactly how it works. That ventral firewall is a transcription factor called engrailed 1, or N1. In direct contrast to the dorsal side, the ventral ectoderm expresses N1. The specific critical job of N1 is to actively repress and inhibit 17A. Yeah, by inhibiting it, N1 restricts the actions of Want7A strictly to the dorsal ectoderm. It holds the line in that turf war, ensuring the palmar surface remains completely palmar.
And when that firewall fails, or when the Want7A signal breaches the line, we see very specific, devastating clinical presentations on the wards.
Unfortunately, yes. A failure of these antagonistic systems produces severe anomalies. Depending on which side of the molecular balance is lost, a surgeon might see duplicated palms where dorsal structures fail to form, or conversely, circumferential fingernails wrapping completely around the digit because the N1 firewall failed to stop Want7A.
Let's take a breath and synthesize the highest yield takeaways for everyone listening.
All right, so we reviewed a remarkably fast biological timeline, from the initial appearance of the lateral plate mesoderm bud on day 26 to a miniature, fully separated adult limb by week 8.
Right.
We tracked the shifting dominance of the limb's vasculature, notably the median artery starting as the primary supply, then being replaced by the ulnar and radial arteries around day 44 before regressing to serve only the nerve.
The starter highway.
Exactly. Finally, we defined the 3 crucial coordinate axes: the proximodistal axis governed by the AER and its FGFs, the anteroposterior axis governed by the ZPA and its sonic hedgehog concentration gradient, and the dorsiventral axis dictated by the non-AER ectoderm through the critical balance of dorsal 17A and ventral engrailed AC1.
It is an incredibly delicate sequence of events. I want to leave you with one final thought to mull over as you continue your board prep. When we study embryology, we spend so much time focusing on the creation of structures, the proliferation of cells, the extension of the limb bud, the formation of bone.
Oh, absolutely.
But consider how much of a normal, functional human hand relies on highly programmed, deliberate destruction. If a simple graft of ZPA cells in a chick embryo can induce a perfect mirror image duplication of a limb, consider how intricate and delicate the timing of sonic hedgehog expression must be in the human embryo, and how easily a slight molecular hiccup could manifest as the complex congenital hand anomalies you'll be seeing in the clinic. If the median artery doesn't regress perfectly, or if apoptosis doesn't aggressively carve out the exact right amount of negative space between the digital rays, the entire architecture is compromised. Congenital hand anomalies are just as often a failure of the body to destroy what it no longer needs as they are a failure to build something new.
That's beautifully said. It shifts the perspective entirely and perfectly illustrates why understanding these foundational embryonic mechanisms, both the building and the sculpting, is non-negotiable for reconstructive practice.
That is all for this review session. If you have a comment or question about anything we unpacked today, drop it below. Thank you for listening. Keep grinding on your exam prep, and do not forget to hit that follow button so you catch our next deep dive session.